I want to welcome everyone to the Bangalore Life Sciences Cluster Science Cafe. We used to do this offline. It was one of our favorite ways to engage with people outside of the campus. But unfortunately, because of the circumstances, now we've been forced to go online. But not to worry, I think we have a really, really interesting session up today. I want to tell you a little bit about the Science Cafe. The Science Cafe is supposed to be a way of breaking away from your regular science talks. So this is not going to be your regular science talk. It's not going to be a bunch of scientists just kind of speaking endlessly and then you have to wait your turn. This is supposed to be disruptive, it's informal, and it's casual. And the point is here is to have a conversation. So this session is entirely dialogue driven. We're not looking at monologue. So if anybody in the audience has a question, please put your hand up and we will admit you into the room and you can ask your question to whoever you'd like. We have really interesting people with us today. Last but not least, we have Taslim with us who's the director of CCAMP, and he's going to be leading the conversation on innovations on COVID-19. I'm now going to give it over to Debarshini, also from CCAMP, and Debarshini is going to introduce the panelists. So over to you, Debarshini.
Hi, hello.
Go ahead, Debarshini, we can hear you.
Yeah, 1 second. Yeah, so hi friends, welcome to today's BLiSC Science Cafe webinar, CCAMP COVID-19 special. Most of you are aware, but for those who are new to this, Center for Cellular and Molecular Platforms, or CCAMP, is a part of the Bangalore Life Sciences Cluster, BLiSC as we call it. CCAMP is one of India's most exciting innovation hubs where we promote cutting-edge research and innovation in the life sciences. We enable deep science or deep tech ideas to reach society and create change where it's needed the most. Coming to the COVID-19 pandemic, the pandemic has clearly emerged as one of the gravest public health challenges in recent memory, with far-reaching implications for entire human society, for the BLiSC Science Cafe, as well as Mahin just told you. And as we speak, India has registered over 2.5 lakh cases with 8,000 plus fatalities. Life for all of us, or maybe some of us, may never be the same in the post-COVID era. But where there is a will, there is a way, as CCAMP and its band of innovative thinkers and entrepreneurs have shown us in the last few weeks. A number of high-impact innovations developed in India can potentially change the COVID-19 game, and today's webinar is about highlighting those efforts. In conversation with us is Dr. Taslimarif Saiyed, CCAMP CEO and Director, who will tell us how CCAMP swung into action in March itself when the pandemic was not at its peak and launched a CCAMP COVID-19 focused Innovations Deployment Accelerator, or C-CIDA. And then joining him in the discussion will be Nitish Kumar Jangir, co-founder and CEO of Koyo Labs, Mayur Shetty, co-founder and CEO of Black Frog Health, Dr. Jogin Desai, founder CEO of iSTEM Research, and Akshay Singhal, founder CEO of Log9 Materials. So request you to all stay tuned and locked in, and requesting Dr. Taslim to tell us more about CCAMP and its COVID-19 innovation efforts.
Thank you, Debarshini, and also thank you, Chandrakant and Mahin, for organizing this particular webinar. It's a pleasure to connect here, and also an opportunity to share with the audience today, our friends, about some of the work that has happened right from here, more or less, from our surroundings. As you know, CCAMP has been set up as a catalyst or an enabler of cutting-edge research and innovation. And what it has been doing over the past few years is to support early-stage ideas which are risky, which are brave, bold, and see if those have any application in society. And that has been a broad, and it has been a broad effort, and it has resulted into a phenomenal outcome in terms of some of the startups are now globally recognized as the leaders in their respective fields. While we are doing this, come COVID, and as Debashini rightly said, around March, we realized that we didn't have enough resources to actually tackle this pandemic in India, be it for testing, be it for support with the ventilation, be it support with sanitization, be it with help with the repurposing of the drugs, be it with sample transport. There was a lot of challenges we didn't have, and many places, of course, didn't have ready-made solutions. So at that time, CCAMP, along with its phenomenal partners like Social Alpha, MedTech Connect, UN Health Innovation Exchange, PATH, and many more. I'm not able to take all names. CCMB, AIC CCMB, Zintio, and several more who actually come together and launched this deployment accelerator with a clear mindset saying that let's kind of see if we can harvest the innovations which are right in our backyard and see some of them are ready to be deployed in a month or two, because the requirement was actually in a month or two. It was not starting a new research, it was deployment that had to happen in a month or two. So we launched this around last week of March, and we got phenomenal response, of the gigantic response. We got nearly 1,100 innovation submission within a month, and with the help of a lot of experts on in CCAMP, some of the really exciting colleagues that we are blessed to have at CCAMP, and of course partners, we were able to assess these innovations with their potential to make an impact once deployed. And within a month, we picked 30 innovations which we thought were the stars for impact, which were the really the best potential to make an impact on the ground for all of us to fight against COVID-19 pandemic challenge. Happy to share that out of this 30, 18 of them are in the field now. That means they are serving humanity around us. It may not be at the scale that we require, but it is actually in the field. And we want to build that further together. So today we have a chance to actually get a glimpse of it through some of the friends we could request and who are with us to share that story or the journey, what they are doing. And I think this is really exciting, gratifying that India, which is not necessarily known to build healthcare innovations, we have examples right here where innovators have built their products and brought it to market or bring it to the labs where they can be used at the final stage to be, for many applications. So with that, I would say that the today's while we have a larger panel discussion here, I think complete focus on these innovations, and CCAMP is delighted to facilitate and support these innovations and do the catalytic effort in that. So may I begin with starting with the order that Debashini used. I'll use the same order to have a 2 minutes introduction by each one of them about themselves and the work that they have done, which is related to COVID-19. And then we can have informal question answers. And then, if there are inputs from the moderators for the questions that we need to take from the chat box or otherwise, we can do that as well. So let me begin with, I think, one of the, of course, star of CCAMP innovation, Nitesh from Koyo/InExcel Lab, who have done phenomenal work in the ventilator space rather, in terms of building that. So Nitesh, if you can quickly tell us about yourself and the work that you have done. I'm very excited that you have been able to roll out a non-invasive ventilator already, and which is being taken by industry and so on. So over to you, Nitesh.
Yeah, thank you, Debashini and Taslim for this invite. Are you guys able to see my screen?
Yes, Nitesh, we can see your screen.
Yeah, perfect. As Taslim and Debashini, everyone has said that COVID-19 has put us in a spot where we can't go ahead without innovating and we have to change our system in such a way that we can deliver better healthcare at low cost. But it's not just better and low cost, but it's about delivering these innovation at faster pace also. uh, I'm Nitesh. I'm a co-founder of Koyo Labs. We are now a division of in Excel. We are Bangalore-based medical device company. Our focus is specifically in the field of emergency and critical care, and we are working in this space from last 5 years. Uh, Before COVID, we had two products. One of them was them is WebCare, which is an intelligent secretion management system, which is world's only system which automatically senses and removes saliva and secretions from patients who are on ventilator, to make sure none of these infected saliva and secretions reaches to the patient's lung and infect them. So in easy way, patients who are on ventilator, they cannot swallow their saliva anymore. And because of COVID, you might have heard that many of these bacteria and viruses live in saliva. And if it reaches to the, if we don't remove them, these reaches to the patient's lungs and infect them. Our system can automatically sense and remove all the saliva and secretion from multiple locations. So this is a world's only device. We have patents granted across the globe: India, US, Europe, Japan, China, and other countries. This product has already received US FDA regulatory approval from European Union, which is the CE certification, and right now already serving patients which are COVID and non-COVID patient. But specifically, when I talk about what we did for COVID, and Taslim has been, or overall CCAMP has been a really enabler for that part, is a device which we had earlier called SANS, which was a neonatal CPAP machine for the looking at the COVID and looking at the breathing support needed for the COVID patient, because it's a respiratory infection which leads to respiratory distress, where patients are unable to have enough oxygen in their blood. So all of you might have heard about ventilators and need of ventilators for COVID patients. But the recent data which is coming out is saying that 80% of the patients who are going on invasive ventilators are losing their life. So the ventilator is playing its role in 20% in 20% of the patients. Uh, so this kind of scenario, we look at more data, and they found that everybody's saying that we should use non-invasive ventilation, which can save 50% of these patients. But the major issue with non-invasive ventilation is it's open. So when patient sneezes, coughs, or even breathe with the saliva and secretion, they infect the whole environment. Especially caregivers are in really high risk when they are near these with COVID patient with high viral load. And these are the patients who have who require breathing support. So looking at this scenario, we upgraded our neonatal CPAP to an adult CPAP, along with a new interface for the CPAP, which covers the patient's face itself, and it isolates patients from the environment. So this provides the pressurized blended air oxygen to the patient. So patient is still receiving the oxygen support or breathing support, but now they, even if they sneeze, they puke, or they cough, the whole saliva and overall bacteria is contained inside this hood itself. So there is no chances of caregiver getting infected. So this is the product. It's on. We have done some patient trial just now. You can see what one of our colleagues wearing the helmet in the picture, and in the left is patient's right is our device. So, and we are hoping that by this month end we will be ready to launch this product, and we have already started getting inquiries from multiple uh, uh, state governments in India to give this device to them, because clinically almost everyone is saying that they know CPAP works, they know this device can help them, but right now nobody in Asia is producing it. We are the only one, and hopefully we should be able to help patients specifically in high COVID COVID centers like in Mumbai, COVID centers in Delhi, where number of patients and load on ICU has increased drastically. Thank you. I'm open for any question.
Thanks, Nitesh. I think you combined a few things together. And I think just, if I may just add, I think Koyo Labs, CCAMP is very proud to have Koyo Labs with us, have actually what you would call it as, built three important devices. I would just segregate them. One device that you had last mentioned, so I'll start from there, is something called CPAP helmet, which is approved by US FDA for treatment in COVID. And you are building with a big industry. I will not name them, as you have not, but hopefully that will come to market. And the scenario that we are seeing right now in Bombay, Delhi, and other places, this will be able to serve a lot of people, may not require direct intubated support, and hence you will also, save ventilators for others' use. So that's very important. We are hoping that this will work. One important other thing that actually Nedesh just mentioned, another device that is US FDA secretion device, which is related to ventilator. As he said, ventilators also cause secondary infection because of the exposure that it provides to the lungs directly, because of the secretion going there and causing pneumonia, which actually statistics suggest a very high level of mortality happening because of it. And it should, if I'm not sure, but a lot of deaths that is happening on ventilated patients, even in COVID-19, could be because of secondary infections. They have a device which is US FDA approved, which can actually prevent secondary infections. They have done the data, and that is already saving life from that. And the third, of course, is a neonatal CPAP device that you have. So all in all, I think you have a phenomenal package in terms of respiratory support capabilities, where you have the capability to save lives. And the best thing is this: there's not a concept; these are products in market. So kudos to you, Nitesh, for that. We are very proud that we have built this together, that you have been with us for a long time. There are a couple of questions, but I'll come back to it. But I'll take this time and to let you take some breath, and then go to now Mayur. Mayur, who is CEO of Black Frog, doing fantastic work. Well, he actually had a beautiful, beautiful story in terms of using mechanical engineering to see how to build a device that would serve last mile connectivity of cold chain for vaccines. That actually had a big issue. Gates Foundation and others, many others, including government agencies, have put a lot of money in building what you call it as vaccines, but they actually are ineffective if they are not given cold chain through and through. And the last mile of that has been a large challenge. And Mayur, along with his colleagues at Black Frog, have now built that. And we are very proud that this something again has come out of our own shores, where they built it. But come challenge of COVID, how these smart innovators have repurposed that for something that is very, very important in terms of VTM and other capability. So Mayur, over to you to share something about yourself and about your work.
Hi, sir. Thank you for doing this. I'll just briefly touch upon relevance in COVID-19 specifically. So since the onset of this pandemic, focus everywhere has obviously been diagnosis and isolating the patients. But testing has three aspects to it. First one is the collection of the samples from suspected cases, which has had its own challenges with shortage of swabs, VTMs. But the process is in itself is fairly straightforward and something our existing healthcare systems were quite familiar with. The second aspect is the transport of the samples, and the third one is the testing in the laboratory. Now, all of the attention has gone to the testing aspect, since that's very new and we were very unprepared with. And the transport aspect is something that we completely ignored and taken for granted, because hospitals have been using ice boxes or gel packs to maintain the right platform for moving COVID-19 specimens. The fact is that the boxes don't in fact maintain the right temperature and conditions for samples, and they're very dependent on the outside temperature. I mean, it acts one way in summer and differently when it's colder during monsoon now. So this creates huge problems of disturbing that viral load and possibly contaminating it too. And we've already had cases of false negatives arising in India due to improper sample handling. So we've developed a portable medical grade refrigerator that maintains a very strict temperature and easy to sterilize for reuse, so that there's no risk of cross contamination. And we use solid state cooling, so there's no toxic refrigerants leaking or motors breaking down, any of those operational issues with that. As you mentioned, we had initially designed the system for last mile transport of vaccines, but we pivoted because of this current pandemic, and we're live in 2 states now, specifically for transport of collected nasal and throat swabs for testing of COVID-19. And now we are working to speed up our assembly cycle so that when the COVID-19 vaccines are finally approved, we are on the front lines delivering efficacious vaccines and improving the immunization coverage as well. So this is what we're doing. Thank you, Dr. Taslim.
Thank you so much, Bhagat. Again, I think very, very exciting because as we all know, the traditional last mile cold chain is largely a blue box with possibly some ice boxes inside. And when they go to rural settings, within 3 to 4 to 6 hours in a hot climate, hot weather, it starts melting. Nobody keeps a log of it, and the entire the productivity of the last mile cold chain actually disrupts. And I think to have something which is 4 to 5 kgs battery operated that can hold the temperature from around 0.1 to 0.2 degrees change and make sure that it actually attains it. It's a huge engineering task for an application that is in healthcare, and very good to see that it is happening. You have been now taken forward for the support by various means. So it's congratulations for that, Mayur. With this, I will go to another star of our work, that is Jogin. Jogin, who is the CEO of a very exciting CCAMP startup, iStem Research, which began its broad work in terms of the stem cell-based approaches for health disorders, primarily to begin with vision disorders, and gone beyond. But interestingly, during this time, some of their parallel work gave actually a segue to make them one of the very few labs in the world which can be a game changer in terms of a tool to support repurposing of drugs, which is required until vaccine comes. We will have to manage and treat our patients until the vaccines come, which is going to be a long way. So how do we test those? And they're very interesting. Stem cell-based platform is actually capable of doing that. So I'll give it to Jogin to say a little bit about the original work of iStem and then about the COVID-19 innovation that you have done. Thank you.
Thanks, Taslim. So thanks for inviting me to this. So we've been in existence for about 3 years, and we started off with therapies for 2 incurable diseases of the eye. One is macular degeneration. The other is a disease known as retinitis pigmentosa. Both are fairly very, very bad diseases, and we've been working towards them. While we were working towards that, we were also working on a disease known as pulmonary fibrosis, which is obviously an end-stage disease of the lung. And we have a experience in creating lung cells even before the crisis hit. So when this crisis hit, we pivoted quickly and we created an anti-COVID screening platform. And here is why it is important. If you are a lab, first of all, vaccines are going to take time. This is you get all these news saying phase 1 is complete. Now they're going to start phase 2. Then will they start phase 3? At a minimum, the results will come by early next year, and then you will have to roll these vaccines out at a scale that the world has not seen. So the best path for us, just imagine if we have a hydroxychloroquine, but which can actually work. Then your problem is solved, because you have these drugs that have a very, very long safety profile and so on, and you can move those forward they are very cheap as well because there are no patents. So that can be moved forward quite fast. So drug repurposing is, in our mind, the best way forward for the kind of scale that we want. Now, for drug repurposing, what do you need? If you're a lab, you need to test the virus. You need to grow the virus and get the drug and prove that the virus is killed. Because once you get that in vitro, in the lab data, then you can move into animals and then forward. Actually, you don't even need to move into animals because the safety of these drugs is well documented. So then you can really start trials quite fast. So here is, okay, so if you're a lab, if you're a brilliant pharma startup or a company or an academic, and you think there is a particular drug that can work, remdesivir, HCQ, etc., you would need to grow the virus. Now, the problem with the virus is, amazingly enough, it does not grow in the lab. It spreads everywhere, but in the lab, it does not grow. Why? Because it needs the human ecosystem to grow. It needs the system of the lung to be able to grow. If you put it in a dish, it will just die in 2 hours and be done with it. So what we have created is a system of, is a lung cell system, which expresses the same receptors that are expressed in the human lung. So by now you must have heard of the ACE2 receptor. There is another receptor, transmembrane receptor known as TMP-RSS2, which is a transmembrane receptor. So what the transmembrane receptor does is primes the ACE2 receptor to look out for the virus. And as soon as the virus sees the ACE2 receptor, it uses that receptor to enter the cell. And once it enters the cell, it destroys the cell. And if you take this at scale, finally you see the problems that occur in people, including respiratory problems, etc. So our system, so the point is, we have cultured these alveolar cells or lung cells in this parlance, which very, very closely mimic the lung cells. So we have now partnered with 2 organizations, and more conversations are going on. One is CCAMP in Hyderabad, which is one of the best academic organizations around in India, and they are starting to use our system to grow the virus in a very predictable manner. The other we have partnered is with Purdue University in Indiana. Again, the same concept. There are 3 big researchers in Purdue who are using our system to grow this virus and see which drugs are going to be effective. Purdue has actually gone one step further. They have also done computational models of drugs. So they have done a computational model of remdesivir and the virus, and they are trying to figure out if the interactions can destroy the virus. And they have a live blog, it is online, where they look at probability of drugs working. So according to them, hydroxychloroquine doesn't come in the first 45 top priority drugs. Even remdesivir doesn't come in the top 30. So there is a lot of science that they can produce quite fast if they have access to a system like ours. So our hope is that with our system, we can make a huge dent in figuring out how to do drug repurposing for the virus. As I said, we've started 2 collaborations in the last 3 months, and we are hoping that we will get more and more such collaborations going in the future. So I'll stop there and see if Taslim, this is more or less than what you would want.
I think this is fantastic. It's a very good representation of how different angles of biological, the combination of biological aspects come together and create a platform that will be useful for drug discovery purposes. And if this works, if I'm not wrong, this would be one of the only 2 in the world actually to be able to provide that. And that is a very solid, that will be a very solid aspect.
So as according to our information, whatever we can see, it is us and there is a Boston-based group. These are the 2 people who are ahead of the game, and there are others obviously that will follow. But yes, hopefully we'll be able to make a difference.
Absolutely. So this is very, it's a good zone to be in, where you are actually globally competitive and building something that is cutting edge. And that's very, very important in such times from India to come. And again, it's proud that CCAMP is in the mix of things with you guys and helping and building this together. So that's very, yeah. So it's very, very exciting. Thank you. So with that, I think there are some questions when questions come, please have a look at them as well. With that, I'll go to Akshay. Akshay, who I think the Akshay's startup became very, very famous last 15 days when a big news went out that their one of the product is now has been put up in French president's residence. And while that is definitely a kind of what you would say the highlight, but the Log9 materials, the company that Akshay is leading, is actually a phenomenal material science and nanoscience organization which does a lot of work. And they began in terms of the use of graphene for antimicrobial properties, in terms of biofilm formation. They have many other aspects to it. It's again, as I would say, the integration of the another discipline of science to actually make an important application in terms of broadly healthcare, even for antimicrobial, antibacterial property. Here, they have built a very nice small oven, as they call it. They call it Karuna oven. And I'll let Akshay say a little bit about that along with its as their own company, or what they have been doing for this COVID-19 pandemic in terms of building innovation that actually has a phenomenal use in the society. Akshay.
Thanks, Aslim. Thanks for the phenomenal introduction. Thank you guys. Thanks for having me here. So as Taslim was mentioning, although we work on nanotechnology and we have been using graphene as a material to come up with various innovations on the energy storage side, filtration side, and antimicrobial side. When the outbreak happened, we thought that we should use the resources and expertise of our team in product development and product building, and see if we can do something to basically help in this fight against COVID-19. So that's what resulted in this product called Karuna oven, which is basically a UV disinfection chamber and can sanitize any object that you place inside very quickly. So when we started, it took 10 minutes, but now we have further advanced the product, and it now takes as low as 4 minutes to disinfect anything that you can put inside. So just to give you all a perspective, this has already been deployed at various police stations as well, and it's helping the front-end responders in our fight against COVID-19 to keep them safe and active. So if you look at in Bangalore, every police station has one Karuna oven already available in its premises, and they have been using it to sanitize their mask and reuse them over time, and hence reduce the stress on supplies of mask and other PPE. So overall, when I talk about, one good thing that our team was able to achieve is that we have taken this product from idea to all the way to manufacturing and deployment in the market in flat 15 days. So we started working on this on 24th of March, and by 11th of April, we're shipping out the first units. So far in the last 45 odd days, we have been able to ship out 2,500 plus units. And we have many more large, small variants of this chamber, which have been deployed across various industries. And we are deploying more variants of the same technology. We are the only certified product in the category, which has been certified at multiple levels, including at ISC and CSI by CSI, CSIO, which has been impaneled by ICMR for certifying UV-based equipments. Also, there is a very simple path lab test called biological indicator testing, which is a standard go-to test for checking efficacy of any sterilization system. And that also has been done on our system. Also, just wanted for the interest of the audience here, given we're talking about life sciences. So this is what our chamber has been able to do. First, I talk about the test that we have done. We have been able to do at Dr. Dang's lab. So if you would understand that Candida fungus is a very hard to kill fungus, and also MRSA bacteria is a very drug resistant bacteria. And when we cultured these two bacteria in two petri dishes, and one was exposed to our chamber, it was able to completely neutralize both the fungus as well as the bacteria. Also, one of the path labs in Delhi bought a sample from us in initial days, and bought one Karuna oven from us, and they, for their internal validation, tested to try to neutralize COVID-19 with it, and they were successful in completely neutralizing a full vial of COVID-19 helicode. So that's the kind of efficacy, and with that we have been having multiple deployments in hospitals, and it has been helping doctors stay safe and keep their families safe, because when they go back, they can sanitize all their gadgets, their specs, mobile phones and stuff like that. Also sanitize and reuse their mask and everything. and hence it has been largely adopted across the country in various hospital chains. Another thing that has happened is that we have been able to innovate and further come up with larger variants of this technology and have deployed it at the Bangalore airport for baggage trolley sanitization, conveyor belt sanitization, basically all your hand baggage, check-in baggages, even escalator handrails. So all of this has been innovated and already deployed at the Bangalore airport for large-scale sanitization of entire passenger movement. So that's what we have been able to do. This is the kind of deployment we've already done across the country. And so that's what I had. Thank you. Thanks a lot.
Thanks so much, everyone. The kind of things that I've heard today have really been so eye-opening. And I personally feel really happy to be part of this cluster and know that such exciting work is happening on our campus. So thanks so much, Taslim, also for bringing together all of these really interesting voices. I now want to open this out to the audience. As I mentioned earlier, the science cafes are supposed to be as disruptive as possible. So please, if you have any questions at all, anything at all that you'd like to ask anybody here, any of the guests, please put your hand up. I'd be happy to admit you into the room. You can also ask Taslim any questions you would like about CCAMP itself and some of the other work that CCAMP does and the kind of the other kind of innovations that CCAMP is spearheading. Remember that COVID is something that we're dealing with right now, but it's not always going to be the case. Hopefully, there are many other things. So this is a great opportunity for you to interact with Taslim since he's here and ask whatever questions you want to Taslim as well as anybody else. So on that note, Taslim, I'm actually going to ask you a question. So Parumita, they asked a question really early on, and I don't know who to address this to because it's a really big question. You know, she's saying, do you think it's the right time to imply, I think she means implement unlock 1.0?
Yeah, so I think if I read the question, if I'm not wrong, what she meant is that, is it the right time to, relax the lockdown and so on? It's a policy aspect that people are looking at in terms of, but in pure play infection spread, it is not the right time. The reason is that I think if you just put the graph and see what we are moving up, our numbers are steadily increasing and there's nowhere near the plateauing. So we are not even reaching the peak, after which we will see the plateauing happening and then the decrease would happen. The projections go that possibly June and July would be the one, rather late July would be the one where we'll see the peak. So if you just put the curve and keep going up as more testing become available, as more tracing happens, as more kits are available, I think our numbers will possibly surge a bit more. So if you are from the infection spread wise, the lockdown is going to be useful in terms of keeping it tight. Policy level decisions because of economic and other aspects of things may be a different reason. But yeah, the more we keep ourselves very, very safe and limit our exposures, it's good for us. I hope that answers the questions.
No, in fact, I think that what you've had to say is really important for all of us to hear. A lot of us, people who are from the scientific background and people who are also not from science backgrounds, these questions are constantly part of conversation today, right? I mean, you can't really have a conversation without COVID entering into the conversation. And the Science Cafe that we've had today has really shed light on some of the things that are happening in terms of the innovations. But this question, I think, was something that people wanted to ask, and I'm really glad that you were able to answer it. There is another question here. Mandrakar asks, in the processes of making the test kit by NIV Pune, he says, Dr. Priya Abraham grows the virus in a lab. Do we have any idea how she did it? Taslim, I think you're muted.
Sorry. So I don't think so. We have insights or context to the details here. Yes, NIV has apparently come out with the kit and it has been also licensed to a few companies. Do not have details of who has the
Yeah.
Can
So I think maybe you need to.
Can I attempt to answer that?
Yeah, that'd be excellent. Thanks so much, Akshay. Go for it.
Yeah, so basically, I think it's not like growing the virus is impossible. It is definitely possible, but the real concern is that do you have the necessary kind of precautions and necessary safety measures available in your labs, and is it the right kind of facility for handling such an infectious virus? So I think NIV is one of the places in the country which has that kind of facility, which we call BSL-3 and above facilities, and they have that kind of I would say approvals with ICMR and the other bodies that they can do such kind of viral culture. So it is possible, but there are a lot of constraints in terms of how do you do it, the difficulty levels, and the biggest concern is the safety concern. So as long as you can safely I would say culture the virus with the human cells and stuff, I think it is possible, and that's what probably they would have done.
Okay, great. Thanks so much. Actually, that's definitely given us some insight. That's fantastic. We have a question from YouTube. As you know that we are also live streaming to YouTube right now, and there's a question from someone there who's asking, how many times can the same PPE be sanitized in the Karuna oven?
Okay, so as per various studies that have been conducted, so there is no problem in sanitizing a mask for as high as 50, 60 times. But the problem really is that when you are sanitizing it, so even if you're doing it daily, for example, after 15, 20 days, by use, there is a lot of wear of the mask, and typically people don't do it more than 10 times. That's what we have seen. So 10 times what people are doing, but in terms of efficacy of the mask, or in terms of structural integrity of the mask, or the maximum number of times you can do it, there is no restriction even up to 50, 60 times. But in practical sense, people have been doing it for not more than 10, 15 times.
Okay, okay. Paramita, you put a question in the chat window. Do you want to ask the question yourself to Nitesh? Would you like to ask the question? We can. You're in the room now. Maybe you can address Nitesh directly. Paramita, are you there? Hello, hello.
Hello.
Okay,
Hi.
yeah, here you are. Hi, Paramita. You wanted a question. You had a question for Nitesh. Do you want to ask him directly?
No, it's okay. Anybody can answer. I mean,
okay, anybody can answer. But why don't you ask your question?
Okay. So, can I ask now?
Please do.
Yeah. Actually, I would like to ask that in India, such a big country, do you have that much infrastructure to accommodate each and every COVID-19 patient? Hello?
Did everyone hear the question?
Yeah, I think the question was that, that do we have infrastructure to support each and every individuals in the country?
Yeah.
Yeah.
Is that correct?
Yeah. Yeah. Yeah.
Yeah. So I think anyone wants to respond to that? If in the interest of time, I'll just take a first, and I'm happy to be supported by anybody. Largely, I think you're seeing that across the globe, people have struggled because healthcare infrastructures are not meant to, possibly be ready for pandemics where
Of course.
such large, numbers and scale happen to be severely impacted and ill, and a lot of them will go in the dedicated zones where infections are there, ventilators are needed, and
Yes.
other things are there. Yeah, no, this is a completely different scenario, and that's why a lot of countries are actually not done as well in terms of treatment. Of course, one is infection control, but even treatment, which has resulted to a high number of deaths per se. We do hope that India would be able to do it. Rather, the first hope should be that we will not need something like that. The hope is that we will limit the infections. We will ensure that if early infections happen, we trace them early and ensure that we can take care of them rather than letting them go all the way in terms of need for so many ventilators and so on and so forth. So that is where I think would be, but in hotspots, possibly like Maharashtra and other places, where the intensity of infections would be high, the intensity of the patients would be high, it may be difficult. it may be difficult. I just saw a note from Wipro. They have converted their entire building to a hospital where patients can stay. I think many other people have done it. This is adding to the infrastructure, right? So that's where it is valuable to make sure that the infrastructure is ready in hotspots and ensure that you are ready to do that. Anyone else want to respond? I just took a first step on it.
Excuse me, sir. I would like to ask one question. That if we don't, I hope that we do not need to reach the mass in that manner. It can be prevented at the first stage only. So if thing happens that we need to reach the mass, then can we reach the mass with that ventilator? Do Nitish Kumar, Dr. Nitish Kumar Jangir and his team has any plan to do that? Hello.
Hi, Nitesh. I think Paramita is directing the question to you. Nitesh, are you here?
So till Nitesh comes in, I think we
Okay,
will,
fine.
yeah, we will need
Yeah,
a lot
sure.
of, yeah,
Yeah.
we'll need a lot of this. Yeah. There is a question that I wanted to share on the question and answer. Question is from Aparna K. Mohan, and it's directed to iSTEM. Was the virus cultured on the lung stem cells in the lab as a positive control? And that is to you, Jogin.
Yes, thanks. So the answer to that is no, Aparna, because you can't, because to get access to the virus, there are very, very few academic labs in India, because you need to, the virus has to be under very, very strict control. What we did do is make sure that the lung cells that we have are very similar to the native lung cells with a few things. One is we grew them on an air liquid interface. So you could see the basal cells and the ciliary cells on top. So you can see the exact way the lung cells are structured, and we could see that they were growing just like that there. Second thing we did is we made sure we checked for the specific receptors, the ACE2 and the TMPRSS2, so a variety of means PCR being one of them, and we proved that these lung cells have a massive amount of, that we have a significant amount of ACE2 and TMPRSS2. This is, the CCMB and Purdue are doing this exact same experiment that you're talking about, which is, I think somebody else asked how is NIV growing the virus. It is probably on the Vero cells, which is a monkey cell line, which is the closest right now available for growing the virus. So our length, hopefully the hope is that, so CCMB is also doing the same thing. They're growing it on a monkey cell line. And now they're going to shift in the next 1 week to the human cell line, which we have provided. So hopefully there we will be able to use the Vero cell line as a control and figure out if the human cell line is better. Theoretically, biologically, the virus should grow much faster. But as in science, we don't know when until we actually see the data.
Great. There was another question. I think, Akshay, you were going to take the question?
Yeah.
Go for it.
Sorry, yeah, I will do that. So basically, the question was regarding usage of Karuna oven in areas of higher footfalls. So basically what we have done is, the box that you saw in the images, that is one, definitely it can be used. For example, the question was particularly about schools. They will reopen, and then kids will be coming in, and they will be scared, and obviously parents will be concerned that the kids don't catch the infection in the schools. So obviously in terms of sanitizing the mask, or basically the stationery in the schools and stuff, Karuna oven can. Beyond that, there are other interventions that we are currently working on and we hope to deploy soon, which is in terms of sanitizing air in the enclosures, in the enclosed rooms, by using UV technology. And so we are coming up with those kind of systems which can ensure that the viral spread in an enclosed environment can be minimized. So those kind of systems are coming up. The follow-on question on the same thing was around what were the costings and stuff. So I think for a typical school, if you have like around 8 to 10 of these Karuna ovens, depending upon the school size, obviously, but per standard, if you have one Karuna, one for disinfecting the items that the students might be using or teachers might be using. I think that should suffice, and the product starts at 11,000 rupees, and depending upon size and volume, it goes all the way up to 23,000 rupees. So you can choose as per the size of the school, and it's easily available on our website, karunaoven.com. But talking particularly about enclosed environments, I think our interventions in terms of enclosed environment air purification, particularly targeted towards COVID and airborne pathogens, is something which will come up very soon.
I hope that answers the question. We have a question from an anonymous attendee. I think this is a pretty important question, and it might be directed to pretty much everybody in the room. And the question is, how well are we prepared in case of a second wave in terms of innovation development and deployment? So maybe Taslim, do you want to take that?
Sure. Yeah, I think it's an important question. I think it is an important question for many reasons. While we are still, come to terms with the first wave, I think as people are, assessing whether there'll be a second wave before we get vaccine and so on, we need to continuously work. We cannot possibly, let our guard down. And I think it is true for the innovations as well. I think while the ones innovations like from the Koyo or the Black Frog or Ice Stem or Log9 materials, where they have been very different kind of innovations, we'll need more of them. And very importantly, for the second wave, we need to be the scale of their deployment also be done, because we didn't get time. A lot of these innovators did not get time to actually productize and scale it up. They still don't have the wherewithal to do it at large scale. They are struggling making some units and giving to state governments to deploy. So I think we need to prepare to see if there is a larger requirement later on. We need to have those things ready. So I would say we need to do that. We cannot just take chance and say, oh, if it comes, we will wake up again and do that. But I think we need to do that. But I think larger challenge would be that whatever we do, we should have scale up of the deployment in mind to begin with, not just the one productization, but scale up of the deployment, because India is a very vast country with such a large population and very different infrastructure. So we'll need to have that going. Like Mayur, if I may ask Mayur to say, do you think if vaccines come, they do have the transport machines ready to take it to all? I don't know what the number, the readiness is. Mayur, what do you think?
Yeah, so I think absolutely that's definitely the biggest challenge right now, which is manufacturability and replicability of the system that we have in place. We've been deploying essentially prototypes that we've developed. We know that it works from a compliance standpoint. We're fine, and when the first outbreak did take place and we had the first lockdown, we were just fortunate that the viral samples required the same temperature range that vaccines required. So we pulled all of our engineers back and we just used all of the component that we had in place. We assembled it. The supply chains were cut off. We had no way of manufacturing more than what bill of materials that we had in inventory. So that is certainly going to be a challenge now moving forward, figuring out the protocols and manufacturing practices so that we are in a position where we can deploy not 10, 15, but hundreds and thousands at a time. But I think one of the key aspects of this, or looking at the bright side of things, is that what COVID-19 has kind of brought about this sense of scientific temperament among all levels of government and people, which is a very beautiful thing. I mean, it starts from something as simple as the importance of washing hands to everybody asking vaccine kab aa raha hai, which is just beautiful. And I am hoping that we'll be able to ride on this even not just for facing further challenges, but beyond COVID-19 and other challenges as well, where everybody has this respect for science and technology and new technology coming and solving sociological problems, right? Societal problems. So I think that's the first aspect of the problem. But again, specifically answering what you had asked, you're right. I mean, manufacturing in large volume is certainly the challenge that we are specifically facing, and we're trying to work with a few companies, larger manufacturing firms, so that we're exploring some licensing options as well. But yeah, I think you're right. That is the concern here.
Great, thanks. I mean, that was, I think that's a lot of food for thought. And I really liked what you had to say about that, Mayur. I mean, I think you brought in a certain humanistic aspect, which I think we really have to keep in consideration, given that, yes, we're talking about science, but at the end of the day, we're all people and there are these concerns. So we're almost at the end of our session, but we do have one more question. And this question is directed to Nitish. Dr. Arunima Bardhan is asking the question, and she's saying, I would like to know, in your device, is there any collector attached to it where the saliva or other secretions are collected? Is Nitish still here, or has he stepped away? Or is there anybody else who could answer this question instead of Nitish?
I'll just answer. So this is
Sure.
this is the device where CCAMP was heavily involved. So it's largely a simple, very smart device that they have made. So in next to the the ventilator tube, they have actually built something, what you call it as a small catheter-like device, which actually goes towards your trachea, and the secretions are actually taken out, aspirated out, and they go to a collector route right down next to the patient and gets bleached. And bleach is a standard technique in a way to actually inactivate most aspects of it. So what they are doing is that regardless whether it's infected or not, anything that goes out through the secretion actually gets bleached right there in the collector. So they have small thing there which gets bleached, and once bleached, they can then do a proper disposal of the body sample and then take it from there as per the hospital environment. So yes, they have that built in.
Great. I hope Anuma, that answers your question. Devarshini says that there's a question from you too. Devarshini, do you want to ask the question?
Yeah. So somebody is asking how we all know that the remdesivir drug has gone through clinical trials, but someone is asking how is it useful? What is its working principle?
Jogin?
Sorry, I was on mute. The basic principle is that it is already used as a broad antiviral even before. So what they've tried to do, because everybody is in a rush, they've essentially used the same drug and assumed that the same drug will work on other COVID-19. So the remdesivir has been used for what we call the enveloped virus family, and it has been in use so far. So the working principle that they have made is that the same mechanisms of action that are against one enveloped virus will work against another enveloped virus. Typically, this is not the way. I mean, if they had more time, they would go through a much more detailed mechanism of action. They would go through a computational model and so on. But since there is no time, it is a reasonable hypothesis. It's not unreasonable to make that hypothesis. And we will find out. But as I mentioned in the earlier, when I said Purdue has done a lot of computational modeling, which is the first thing you would do before going into trials, where they have said that there are lots of other drugs that have a much higher potential than remdesivir as well. And I think if you Google it, it's a blog that is ongoing. It is updating every day based on results that they have. So I hope that answers the question.
Great, thank you so much, Jogin, for answering that question. So we're at the end of our time. If anybody has one last question they want to answer, we'll allow it. But if not, then we will close this Science Cafe. Yes, okay, so Mandrakar has a question. So Mandrakar, I'm going to allow you into the room and unmute you. So why don't you just ask your question directly? Please go ahead.
Hello, can you listen me?
We can hear you.
Yeah, I want to ask that ICMR constantly rejected that there is no community transmission. Every day there is lots of case increasing. How you guys think about it? Because if this number can traced, then this is not community transmission. But I don't think this number can be traced, this large number.
Okay, that's a big question. Does anybody want to take that question?
I'll take a first shot and hopefully somebody can come and back me up.
Yes.
I think Mandrakar largely, yes, it's. I think today and yesterday there was a indication to say that yes, it's not a community transmission. I think these are early indications by different agencies, but just for the normal public, if you want to talk about it, if individuals are not traced completely and are not linked to something that is not a transmission within community, it is difficult to say yes or no about it. But as the numbers are growing, and hotspots are happening, you could possibly argue that yes, there is a very high chance of community transmission as well. So I just thought I'll give you that perspective. You will need data, you will need evidences to actually say this way or that way. But if one wants to have an opinion, you can definitely have the opinion that there are chances of community transmission. I hope that answers your question.
I would like to add a bit to what he had said. I think community is a very vague term. So, what do you mean by community? Because a small locality in a city or a city are two different types of community. The nation is a different kind of community. And when we talk about community transmission in a country like, for example, Italy or Spain, they are much smaller countries, probably almost half the size of many states in India. So, from that perspective, a community spread happening over there is a nationwide community spread, whereas what is happening probably in Delhi and Mumbai can be termed as community spread, but not across the country. So there are various states and districts which have not seen any case in the last few weeks. So it's not a nationwide spread, I would say, but definitely there are early signs of some sort of community spread happening in hotspot areas across the country.
Great. Thank you so much, Akshay, for that input. And with that, I think we have to come to a close of this Science Cafe special with CCAMP and the Innovations for COVID. I want to thank everybody who's made the time to be here today. Taslim, Mayur, Akshay, Jogin, Nitesh, and also Debarshini. Thank you so much for being part of this. And please keep your eyes open for other science cafes that are coming your way. We will continue to be online. And please remember to spread the word and stay as interactive as possible and find out about what science is going on. So thanks again, everybody.
Thank you. Thanks so much. Bye.
Have a good
Bye,
weekend.
guys.
Thanks.
Yeah. Thank you all.
Thanks, Mike.
Okay.
Bye.
Closing the meeting now. Bye bye.